So as a next speaker, I want to hand over to Julia Philipp. She's in the consortium GHGA, and we're probably going to learn in a minute what it is all about. She's from the EMBL Heidelberg, and her slam is going to be about sometimes your games ask for sugar. Let's see what she will tell us.
Yeah, thank you, Sonia. I'm working on sharing my slides. Is this working okay?
Yes, perfect.
Okay, thank you. Yeah, so like Sonia said, my name is Julia Philipp, and I am from the German Human Phenome Genome Archive. That's what GHGA stands for. And we'll talk about this in a minute. But yeah, I want to start with a story about why sometimes your genes ask for sugar. And no, I will not tell you to eat all of your leftover Halloween candy in one sitting. Instead, I want to talk about Sarah and why her doctor told her to eat 3 spoonfuls of sugar every day because of her genetics. But let's start at the very beginning. This is Sarah. She's a young and very real girl living in Salzburg with her parents and her sister and her fluffy cat. Around the age of 4, her parents noticed a delay in her mental development, an unusual round face, and a delayed growth compared to her peers. And after not being able to find out a reason for her symptoms, Sarah's parents finally took her to a specialist. The specialist decided to test for so-called rare diseases, since none of the common tests resulted in any answers. Rare diseases are diseases which affect less than 1 person per 2,000. And while a single rare disease does not affect a lot of individuals, a large number of people are affected by one of the up to 7,000 rare diseases described so far. In Europe, that means about 20 to 30 million people are suffering from a rare disease. And around 80% of these rare diseases have a genetic origin. And for that reason, the specialist that Sarah visited decided to draw some of Sarah's blood and have her genome sequenced and hopefully find out what's making her ill. But of course, Sarah was wondering, what does genome sequencing even mean? And most importantly, is it going to hurt? So the specialist explained it to her like this. Our genome is tucked inside the core of each of our body cells, like it's in a little safe, and we can find it inside of our skin cells, our hair follicles, or our blood cells, which is why she took Sarah's blood. And if we zoom into the core of these blood cells, we can find the genome. It's usually like a long thread, and sometimes we can find it wound up in these little spools. Now, if we look closely to these little spools, we can see that is actually a long intertwined thread, which we call DNA. It's mostly made out of 4 different building blocks that scientists abbreviate to the 4 letters A, C, G, and T, just to save their time. And the doctor told Sarah to imagine that these 4 letters in the DNA kind of work like a code, like Morse code. So stretches of this code encode messages that are called genes, and the entirety of our genes would make up our genome. And this code determines many of our traits. And while 99% of this code is the same in all humans, the last 1% can differ quite a bit between people, causing us to have different traits. So for example, this can encode our different eye colors, blue, green, and it can affect whether or not we have to sneeze when we look into the sun. It can even affect the choice of our partners. And also, interestingly, it can affect if our pee smells funny after we ate asparagus. Unfortunately, these small variations in our DNA can also lead to disease. So sometimes as little as 1 letter change can have an effect. For example, it could cause the lack of the enzyme lactase and therefore cause lactose intolerance. These little differences can also cause cancer, or as we're suspecting in Sarah's case, a rare or unknown disease. If we don't have medical tests available to diagnose a patient, we can try reading out the entire genome in a patient's cell to find out what's making them ill. So in that case, a lab scientist will just feed the patient sample that contains the DNA into a very magical machine that we call a sequencer that is going to read out every single letter that is encoded in our DNA and send that to our computers. This process is called sequencing the genome. We've briefly heard about this before, and it's a really interesting process, probably a science slam in and of itself, but what is even cooler is what we can do once we have this information. So the machine spits out the letter sequence letter by letter, and since the human genome stores a lot of information, the letter sequence is going to be very long. And even if we're not talking about bacterial genomes, we're still going to have a lot of letters. So if we printed the human genome sequence into books, it would be about 175 volumes. So obviously at this point, Sarah is a little worried, and she's like, well, if my genome is filling 175 books, how are you ever going to find what's making me ill? She has an excellent point here. If we try to read out every single letter and just try to find a single difference, that would take us an estimated 95 years to do so. So we need something faster. And for that reason, we need computers, and we need to develop computational algorithms that I'm depicting with this magnifying glass here that can handle large amounts of data and can detect really small differences for us. These algorithms use reference data from other people to compare, for example, Sarah's genome to a person that is healthy in order to find these small differences that could explain her genome, her symptoms. And so here's just a little example of what it looks like to look at this genetic information. So up here in the top row, these big colorful letters, that's a reference genome. So a person that's considered healthy, or a consensus of healthy people. And then these gray short snippets are what the sequencer spits out. So our algorithms first have to puzzle back all those sequence snippets back into an order, according to the reference, and then find these minute differences that could explain symptoms. So back to Sarah. Her genome has been sequenced, and her doctor, together with a team of scientists, have applied these computational algorithms and found a small change in her genome. And what they found was actually just a two-letter change in the gene GFUS, which encodes an enzyme that makes a substance called GDPL fructose. Because of the two-letter change, Sarah did not have enough of the enzyme in her cells and was not able to produce enough fructose. And fructose, however, has a really important role. It's needed to modify the activity of cells' building blocks. And about half of our building blocks need to be modified in that way to function fully. So the lack of this modification explains Sarah's developmental delay. Luckily for Sarah, in this case, that problem could be solved by a specialized sugar that we can buy in a big plastic tub. So when Sarah is eating this sugar, her body can activate a different process, so going around this gene that's a problem and making the fructose in a different pathway. So as a treatment, Sarah's doctor just instructed her to eat a spoonful of sugar 3 times a day. And by now, Sarah is 7 years old. She has caught up in her mental development and her growth, and she's preparing to go to school next year. So success stories like Sarah's are made possible by the data collected and the algorithms that are developed by scientists. But more importantly, and the speakers before me have mentioned this too, is the sharing of those resources, the sharing of the data, the sharing of the software. It would not have been possible to diagnose Sarah's illness without having reference data to compare her genome to, and without having the algorithms that detect these differences. And vice versa, Sarah's data can now help other people. So scientists who receive her genomic information can work on understanding the disease and find other cures. And clinicians and lab scientists can use Sarah's genomic information as a reference for helping diagnose other patients. By the way, this does not only apply to rare diseases, but also to cancer as well as other types of health data. So in order to enable this sharing, we need an infrastructure where genomic data can be shared, accessed, and analyzed in a protected environment that balances the need for patient data privacy and open data sharing. And that is what we at GHGA strive for. We are working on a data repository that enables fair, secure, and anonymized sharing of data like Sarah's genome and of algorithms that can identify relevant information in these data. Thank you.
So thank you, Julia, for bringing us, giving us some insights into this important research area. I'm very glad that the little girl Sarah is doing well, and your research could help her. And I think this way we can see how important the different research areas we today see, and look at how important this research is, and also how important it is to share the data. And it's also great to see that Sarah is so well that she can enjoy all the Halloween sweets, as my kids also do, and I think that's also very important for her. But we also see in this research area actually how important it is to have a balance between open science and on the one side, and also about privacy, about anonymization, pseudonymization on the other side. So thanks again, Julia.